In this video, we will be discussing transcription, or the synthesis of RNA as seen in prokaryotes. As just a general overview of the steps for transcription, we will see that we go through template binding, chain initiation, chain elongation, and chain termination. We will see these steps illustrated in just a moment. There will be multiple diagrams we look at explaining the processes previously mentioned. DNA is transcribed in the uncoiled section of this structure. Knowing RNA is read 3 to 5', we can label our template encoding strands, with our template strand being the DNA strand that becomes our template for RNA transcription, which is seen as the bottom strand in our bubble and is read 3 to 5'. A coding strand, on the other hand, is the DNA strand which has a base sequence corresponding to the RNA being synthesized, except instead of having uracil present, the coding strand would have thymine. Coding strands are also read 5 to 3'. Now we will simplify the diagram from coiled to uncoiled just for the purpose of making the processes easier to visualize. We can also begin to add new pieces to the transcription puzzle. As you can see in this photo, we have added 3 aspects to this diagram, being the -35 site, the -10 box, and the +1 site, which are all put together to form the promoter region. The +1 site is also known as the initiation site, which is where the first RNA nucleotide is transcribed. The +1 site also contributes to the RNA coding region, which is where the termination of transcription takes place. The -10 site, also known as the ta-ta box, is part of the DNA sequence where it can begin to be decoded. It is also referred to as the Pribnow site, and is called TATA for its conserved DNA sequence being only thymine and adenine. Lastly, we have the -35 site, which with the TATA box creates what are known as the control regions inside the promoter region. Now we see the addition of a core enzyme and a sigma factor, which are found during the process of template binding and chain initiation. These 2 factors attach to the promoter region on the coding strand and combine to create what is known as a hollow enzyme. Now that we have looked at both template binding and chain initiation, we can begin looking at the second half of this process. An enzyme known as RNA polymerase approaches and binds to a consensus sequence and leads the RNA along DNA. This is what begins to produce RNTPs, which are the building blocks of RNA. Now we can begin the process of termination, where we see RNA form a hairpin loop, which is found because of inverted repeats. As you can see with the base pairs labeled in the structure, the base pairs and their complement align on the structure. After this inverted repeat, we see a separate pattern with what is known as a poly-A tail. This sequence is due to adenine residues in the structure and completes the chain termination. After the process just illustrated, we go from having a double-stranded DNA to a single-stranded RNA. Hypothetically, a gene that codes for a specific enzyme which metabolizes sugar lacks a -10 site, or a TATA box. Would the enzyme be able to perform its job if it lacks a TATA box? Let's talk about it. If the structure lacks a TATA box, this leads to a lack of a promoter region, meaning that the core enzyme, sigma factor, and RNA polymerase do not have a place to attach to on the structure. What does this mean? If the gene is under these conditions, transcription is not possible, leading to the enzyme to not be produced and the sugar will not be metabolized.